Hello, everyone, and welcome to the fourth episode of the Life Design Chronicles. My name is Laura Fajín Ribeiro, and I am the Assistant Director of Life Design for the Advanced Academic Programs and co-host of this podcast. This podcast is part of the Life Design Lab at Johns Hopkins University. If you haven't heard about the Life Design Lab yet, our goal is to support and serve all Krieger and Whiting undergraduates and master's students, regardless of postgraduate plans. Through the Life Design Lab, students have access to a variety of resources, such as life design programs and courses, treks, and other experiential learning opportunities, career search and development programs and guidance, as well as networking opportunities and connections with alumni and employers. This podcast is a space for Johns Hopkins University students and industry professionals to have meaningful conversations about life decisions, and career choices, as well as a safe space to ask questions that have been sitting for too long in the back of your head. If you are curious about how others have done it, this podcast is for you. Please use the chat to type any questions you may have for our guest. This will be read at the end during the Q&A. Without further ado, I'll pass the mic to my co-host, MS in Bioinformatics, Swetha Suresh Kumar.
Thank you, Laura, for introducing me. Hi, everyone. My name is Swetha, and I'm a master's in bioinformatics student currently in my third semester and looking forward to joining the industry once I graduate.
So I'm going to be your co-host for this episode on bioinformatics. Today, I have the pleasure of introducing the Director of Software Engineering at Natera, Joe Barrows. Joe, welcome and thank you for accepting our invitation to be our guest for today's episode. So first, I'd like to ask you some questions about your personal background. So I'm curious about your journey in bioinformatics. Like, how did you first become interested in this field and what led you to where you are today?
Yeah, thanks. Very glad to be here. And by and large, my journey in bioinformatics has been a little bit of a twisty one. So I was recruited out of working at an internship at BWR by a diagnostics company called Council in a field sales role, actually. And I was promoting their molecular diagnostics products to physicians in the Washington, D.C. area. And I started getting really interested in the molecular biology and the biotechnology behind those things. And I was traveling a lot of overnights in hotel rooms, this kind of thing. And so I wanted a really, really flexible and reputable program. And so I actually started the master's in biotechnology at Johns Hopkins. And eventually I decided that I wanted to learn to code. And there I was in the program already in biotech. And so I decided that I could kind of dovetail a couple things and switched over to the master's in bioinformatics. And this coincided quite nicely with my move to Semaphore, which was then Mount Sinai. It was the Mount Sinai Genetics Lab at the Icahn School of Medicine. And where I ended up doing a combination of project management and bioinformatics until I graduated from Hopkins and formally became a bioinformatician there.
Wow, that's very interesting. So what do you like to do in your spare time? Do you have any hobbies that you like to enjoy?
I don't have a lot of hobbies. I'll candidly admit that my job takes up a lot of my time. I do also have a young son, so he takes up the majority of the balance. But what's left, I like to read a lot of nonfiction, biotech, and business books for the most part.
Oh, that's really amazing. So could you walk us through a typical day in your role? What are the key tasks and challenges that you like encounter regularly?
Yeah, absolutely. So the first thing to know is that there really isn't a typical day in my role. There's some things that tend to happen recurringly. So we do follow an agile sprint development style for our software engineering of our bioinformatics pipelines. And so there are some agile rituals that happen on a recurring basis, like sprint planning meetings and things like that. We do also have project planning meetings. And so there are projects that are kind of across the sprint structure. There are lots and lots of one-on-ones where I'll have kind of coaching sessions and sometimes be coached by other people within the company. And then I still try to do some amount of technical work. So like reviewing architectural designs and sometimes designing things at the architectural level. A lot of work in Plant UML as a tool that I and a few others have adopted here.
Okay. Yeah. So are there any, what emerging technologies do you find more fascinating or promising for the future of genomics and data analysis?
Yeah, so I will speak entirely personally and not on behalf of Natera in any way for this question. So I'm quite excited about the proliferation of companies working on cloud solutions to bioinformatics pipelining. So when even 5 years ago, there really weren't many options for setting up a compute stack in the cloud. You kind of had to build something yourself. You had to use like a cloud formation on AWS and then like really tune it. Or maybe you could do something on Azure. And now it seems like almost every day there's some new vendor looking to promote software as a service, cloud computing and storage layer. And this is a really exciting thing for the bioinformatics industry because it seems like we're getting a lot of really nice tools in our toolbox.
Okay, so you're saying what kind of cloud computing resources do you think that entry level students should be knowing about?
Well, I think it's really important to have some familiarity with the modern bioinformatics pipeline languages. We particularly like WDL, and I personally particularly like WDL. That's the language that I've used across a couple companies now in building bioinformatics pipelines. I find it's a really friendly way to build out those directed acyclic graphs that we have to build as we build our pipelines. Others in the industry use Nextflow, so that's another great tool to learn about. And I understand that SnakeMake is fairly popular. I think it's less important to know one specific of the three that I've listed, and more important to have at least decent familiarity with any of them and pipelining and so forth, and understand the problems and challenges and benefits of parallelizing your work across the cloud.
Okay, so do you have any personal favorites that you work on?
Personal favorite tools?
Yeah, tools.
Well, like I said, WDL, I really like Docker for containerization. I also mentioned Plant UML earlier. So I really, really wish I had known about Plant UML when I was a student. So this is a free open source diagramming tool that anyone can use for a variety of software diagramming, and it just would have been so much easier to do some of the assignments and draw diagrams and everything else using such a tool. So I really I use that a lot.
Okay, that's really interesting. So switching gears to mentorship and skills. So have you personally had a mentor in your career, and how has mentorship influenced your professional development?
Yeah, absolutely. So I've had honestly quite a few mentors. Most companies I've worked at, I have been fortunate enough to have at least one person that I would consider a mentor. I'm not going to call anybody out by name, but generally it's been something of an organic thing. Like I've tried to always be curious in my career and always try to keep learning. Even 5 years out of Johns Hopkins, I still learn new stuff almost every day, and that's a deliberate thing. And I think that learning attitude will help you find people to teach you, and that's really what a mentor is someone who has a lot to teach you, who you learn from on a recurring basis. And I think there's a lot of, like, I don't know, formalization around that idea now, like this idea that you need to have mentors and things like that. But really, it's something that just should happen naturally if you're curious and find people to learn from.
Okay, yeah. So, like, do you have any advice that you could give recent graduates about finding mentors in the field?
Well, I think that it's it's really difficult to find your first job as a bioinformatician out of your master's program. And in my opinion, this is actually maybe the most difficult thing that you'll have to do in your career growth. And so I would focus your energy more on that pathway of like finding a first job that's out of biotech, that's reasonably related to bioinformatics. And then once you're there, you should be able to identify people who have a lot to teach you and are willing to teach you, and then build those friendships with them, not with an ulterior motive, but just by trying to learn from them.
That sounds like great advice. Thank you very much. So, switching gears a little bit towards the job search. So, how much weight do employers typically give to academic credentials like a master's or a PhD versus practical skills when hiring for entry-level bioinformatics roles?
Sure. So I can largely speak to myself and my preferences for this question. I do hire bioinformatics software engineers, and I've done this now at a few companies. I don't necessarily need someone to have a master's or a PhD. It depends a little bit on the role. So if, for example, we're doing some complex algorithms work, developing a novel variant call or something like that, that's the sort of thing that you want someone with a PhD for. If we're doing more like production grade engineering of existing algorithms, that's something that someone with a master's or a bachelor's degree is going to be more qualified for. And then in terms of skills, I think that it's most important that people who want to become bioinformaticians learn as much about actual coding and have as much coding experience as they possibly can, and really focus on that more than the biology. Because by and large, you're going to learn biology on the job that's specific to whatever company you work at. And the general background that you can get in academic programs is great and valuable and a good starting point, but you're going to very, very quickly go much, much deeper in whatever corner of biology you happen to be in. And so it's not as valuable a use of time when you're in school to try to really learn broadly biology. It's much better to learn about engineering concepts.
So, like, what kind of technical skills that are required for an entry-level bioinformatics job when you come for interviews, and how does your resume get picked? Like, what kind of skills do they look for then?
Well, so I like to see that they've taken like data structures and algorithms, which I know is required on the Hopkins curriculum. I have found that not setting that as a filter does not set candidates up for success in their role. And in terms of actually like the technical portion of the interview, you should be able to write a decent amount of code in your language of choice on the spot with someone looking at you doing this, right? And you shouldn't expect to write something on the scale of your term assignment for a course or something like that. But you should be able to, hack together a for loop, parse a text file, maybe do some command line shell scripting or awk, this kind of thing. You should have that comfort with dynamic development if you want to be successful in an interview process.
Yeah. So do they expect more of downstream experience, downstream analysis, or upstream analysis?
Could you tell me what you mean by downstream and
Like
upstream?
downstream analysis, meaning like once you get the data, you kind of analyze it using R programming. And the upstream is like when the data just comes directly from the sequencer, and then you kind of do a number of steps like
Hmm.
aligning and then mapping, that kind of thing is upstream. So I was just curious what kind of experience is required for which has more importance.
Sure. Yeah, that makes sense. It depends a lot on what kind of role you're going for. So often you'll find that an entry-level bioinformatics position is a lot more focused on analyzing the outputs of an existing pipeline. So more of that, R-based or Python-based data analysis, like you were talking about, knowing how to use IGV, look at BAM files, look at VCFs, really understand a little bit about what you're looking at as you look at short read NGS data. And that's what a lot of places will call a bioinformatics analyst role. For more like a bioinformatics software engineer or maybe bioinformatics scientist, that's when you start to get into what you're talking about, like the primary and secondary analysis, BCL through VCF, and that which of those areas are more important to the company will be what they focus on during the interview. Generally, for my roles, I am leading secondary analysis here, so I tend to prefer to see that full pipeline understanding versus like deep knowledge of statistical analysis of outputs or something like that.
Okay, okay. So like you kind of analyze ChIP-seq data, RNA-seq data, is that how we could say? Or-
Well, there's a lot of stuff that you need to know about as you analyze data in bioinformatics. I, it's hard to generalize a little bit because you have to look at what the experiment is. You have to look at what the questions are being asked, and then you have to think about how you're going to answer those questions. And so a lot of the time it's sort of experiment specific. One good example, there was an assignment where we had to determine if a particular variant had been called in a whole exome sequencing pipeline. And so to get to the answer to that question, we had to look at the actual pileup of reads using IGV because the VCF was a little ambiguous. And so like understanding how to get at the answer to that question is a sort of specific skill that you develop with experience. But that's sort of an example of the type of analysis you might do.
Okay, yeah, that kind of gave a great idea about what we're expecting. So looking at more of career growth and a future outlook. So beyond technical skills, what kind of soft skills or personal qualities do you believe are very valuable for those starting in the field?
Yeah, so the thing to know about being a bioinformatician is that by and large, you're never done learning. And you should really try to internalize that. So, at Hopkins, we have courses on biostatistics, we have courses on software development, we have electives, we have molecular biology, we have quite a few courses on a bunch of different topics. And you're going to find when you get to the company that like maybe one of those courses kind of covers some of what you need to know. And then you need to go much, much deeper in that area. And then when you end up growing in your role or switching jobs or whatever, you'll find that another area becomes more relevant to you, and you have to continue learning in that area. And you'll also find that you'll lose sight of some of the stuff that you used to be good at too as you grow, and that's okay, right? Like, you're not going to be perfect at everything that you have once known how to do for your whole life. It's much more important to have this learning based attitude, and try to look for ways to learn and continue growing, and never assume that you're done with that.
Yeah, that's really great advice. Thank you very much. So, like, how important is familiarity with biological concepts and genomics for entry-level positions? Like, should the candidates prioritize acquiring domain-specific knowledge?
So I think certain types of knowledge of genomics are going to be more valuable than other types. I don't recommend going super deep into, say, miscellaneous rare diseases in the course of your master's program, unless you're really interested in that and really want to work on that, right? It's much better to try to learn things that are transferable. For example, going really deep on how next generation sequencing works at the various major vendors, like learning how Illumina sequencing works, learning how PacBio works, being able to understand what those data look like, understand what the wet lab process behind those data look like, understanding the whole kind of end-to-end process. And, also, like, sort of almost generic molecular biology knowledge is really valuable. So, internalizing the central dogma, really understanding how genes become mRNA, become proteins, understanding, how that can go wrong, how it's regulated, how we as bioinformaticians interact with representations of genes and data, like understanding what a VCF file is, a bed file, all this kind of stuff.
Yeah, so should we also concentrate on epigenetics as well, right?
Um, that is more specific to what you end up going into. And
Okay.
so I will candidly admit that the gene expression course, or the epigenetics course that was on the curriculum as a required course, was a very long and difficult course for me that I ultimately have not gotten that much value out of in my career. So take that as you will.
Likewise. I just finished it last semester, so I kind of get what you're saying. Yeah.
Yeah,
So
it's a very comprehensive course.
totally, I can totally relate with that. So how important is networking, like attending conferences and staying involved in the bioinformatics community for career growth? And like, what tips could you offer to those who are just starting to build their professional network?
So networking is kind of a difficult topic in some ways, particularly in our modern world of Zoom communications and so much digitally done. I think that it's important to attend at least one conference at some point in your career, um, even if you don't plan to go into deep research or pursue a PhD, um, just to understand a little bit about how social they are, how people share research, what it actually looks like, what people are talking about when they talk about going to a conference. Um, and then if you plan to attend more, um, my advice would be to try to publish as many posters and abstracts as you possibly can. The really nice thing about posters and abstracts is that it means that your industry work enters the public domain, which means that, when you're looking for that next job, when you're trying to grow and so forth, maybe you just want to remember what you did at your last company. There are publicly available records that you don't have to be worried about sharing or looking at or anything else. And you have stuff that you can share with prospective employers. So that's kind of my advice: posters and at least attending one.
Yeah, thank you. Thank you very much. I think it was really useful. So, in your opinion, so this is my last question. So, what exciting developments can we expect in bioinformatics and genomics in the next 5 to 10 years?
So I think I already talked a little bit about my excitement about cloud vendors. So I won't go too far into that topic. I would say also that the cost of sequencing just keeps going down, and it's going to eventually go low enough that really interesting and currently unavailable business models will be available for companies that want to do large sequencing based products. So I think we're going to see some really interesting growth there. And then also the regulations are changing a lot right now for laboratory developed tests. If the listeners or watchers of this haven't read the new FDA guidance on LDTs, I would definitely recommend checking it out. It is my personal opinion here, going to make a non-trivial impact on the industry in general, and it's a really nice summary of the way the industry uses a lot of the tools of molecular biology and bioinformatics.
Yeah, thank you very much, Joe. That's all that I have for the podcast, and thank you very much for your time for answering my questions. And I hope that it's going to be, it's going to be really useful for all the bioinformatics students, and the upcoming bioinformatics students. So yeah, thank you very much.
Thanks for the questions.
Give me just 1 second. We have a question in the chat from Megana. Megana is asking, well, she says, hi, could you tell me if it is important to know many bioinformatic tools or technologies in general, or just one or two tools, but in greater detail? I think you mentioned this earlier.
Yeah, so it's a little tricky to split out what constitutes a bioinformatics tool. So if you're talking about programming languages, I think it's quite important to have pretty deep familiarity in at least one programming language. If I had to vote, I would say Python, followed by R. If you're talking about like having a really, really deep understanding of, say, how GATK does variant calling at the expense of knowing how demultiplexing works, or alignment, I think you're much better off having a basic understanding of many commonly used tools in a typical germline or somatic pipeline versus only really knowing how one thing works. And especially, you should have at least a decent understanding of how the lab works and how they produce your data that you need to consume, because you need to be able to communicate with them about their experimental designs.
Thank you for answering. I'm going to ask, Megana, do you have any other questions that you want to ask, or Swetha, anything that came up that you want to know? This is the time.
Yeah. So
I have a question. So I'm recently taking this course called Metagenomics, which is one of my electives, and it concentrates more on the primary processing and primary steps that you are saying, like aligning. And then I was using more of Bash to write my scripts. So like, is it useful when it comes to an industrial setting to like use Bash to run my commands? Or I mean, it's more of the happening field is Nextflow and the tools like that, right? Do people still use Bash scripts to run their bioinformatics tools like Bowtie or BWAMM tools like that?
Sure. So it depends a little bit on what kind of role and what kind of company that you are planning to work in. But generally speaking, you definitely need to know shell scripting even today for almost any role as a bioinformatician. I'm not as familiar with Nextflow like I mentioned, but I can tell you in Whittle, the way that Whittle works is you have like the pipeline with various tasks within it, and each of those tasks will be backed by a Docker image and container. And the way that you then call the code within the Docker is you have a shell command within that particular task of the Whittle workflow. So you have a bit of shell right in your workflow, just in the course of running your workflow. So that's a little bit more advanced, a little bit more sophisticated maybe than what I'm hearing you're describing, which is maybe chaining tools together on the command line and piping the inputs to one another. But that doesn't necessarily mean that what you're doing isn't valuable or appropriate. When you're doing a lot of one-off analyses, a lot of custom stuff, you're not going to invest the time in building out some really robust cloud-based pipeline and architecture and so forth. You're largely going to do what you're doing now, which is a bunch of shell stuff, awk and so forth. So it's good to know how to do that stuff.
So do you think that this would be like a basics for understanding it, and then we could like build on our understanding by practicing it in tools like Whittle and Nextflow?
Yeah, I think that's reasonable. I think a lot of folks who develop pipelines start with something like you're describing, like a shell script that has some series of commands, and then they break up their shell script into steps of a more bioinformatics specific pipelining language. This just really depends on how much scalability and reliability and things like that you really need to have in your pipeline. What I would say is a little more important than this question, though, is to make sure that you really follow good development practices when you do your development. So like one of the things that I wish I had done, which I did not do, was make sure that every single assignment that I did when I was at Hopkins was in GitHub, right? And that I had really made myself practice a disciplined Git workflow when doing all those assignments. Because you would, take that with you, and then you'd have a nice portfolio to show employers in GitHub, right? So that sort of thing is a little more important, I would say, than worrying too much about like trying to use Whittle or Docker. Like you have to have the fundamentals down first.
Yeah, I can totally understand that. Thank you very much for answering my question.
Sure.
Well, so I think it's almost 40 minutes. I want to thank you for joining us today. Joe, I hope you had a good experience in this podcast. This is the 4th episode we do, and we are really excited for having you. I'm so happy that Swetha agreed to participate, co-host. She is brilliant. She's also our graduate assistant for the Life Design Lab graduate team. So yes, we are going to continue working together, and I hope that in the future, if we have more events or opportunities like this, that you feel like you want to join us. If not, this was fantastic. Final thing, follow us on social media at Life Design Lab, WSC, AAP, right? Okay, thank you very much. I'm going to stop recording. And yeah, that's all. Bye-bye.
Thanks a lot for having me.
Thank you. Bye.
Thank you.